And I'm gonna ask everybody to mute your microphones if you could, please, except Allison and Sarah. And then we'll just give everyone just one more minute and then I'll hand it off to Allison. All right, I think you can go ahead, Allison. I'm gonna mute.
Okay, thanks, Leonor. Well, good afternoon, everyone. Thanks for joining us this afternoon. I'm really excited to be able to introduce Sarah. So Sarah is a student in my lab doing her master's degree, and she has a BS in biology and horticulture from Iowa State. And while she was doing her BS, she worked in Sylvia Ciangio's lab. Sylvia is was a soybean plant breeder. And then she also spent a summer working in Darren Mueller's lab. But she came to me a couple of years ago now, interested in doing her master's research. And I challenged her to apply for a North Central Region Sustainable Ag Research and Education graduate student grant, and she was awarded that. And so that went towards her research. And then we just heard a couple of weeks ago that she was runner-up in the Martin Stoner Scholarship for this year's conference on soil-borne plant pathogens. So she will be presenting some of her research at that conference in a couple of weeks. So anyway, Sarah's research is really cool. It's ironic that it's very fits very well with things going on at the moment in today's science, society, social distancing. And so Sarah's going to talk about her social distancing project on corn and rye. So off you go, Sarah.
Okay, hi everyone, I'm Sarah Kurtz. And the main question that I have for all of my projects is, does the proximity of a winter rye cover crop from corn have an effect on corn growth, development, and seedling disease? First, I would like to give a background on cover crops and corn. The corn and soybean rotation is the most common field crop production system in Iowa. And with that, in the fall and the winter months, the fields are fallow, bare, nothing is grown on it. And this leads to erosion by wind and water runoff. It can also cause nitrate leaching into surface waters, and this leads to environmental contamination. So we need to find a problem for this solution, or a solution for this problem. And one solution that we have found is that is to use cover crops. And cover crops are crops that are not harvested, but are specifically grown to improve soil health. Some of the benefits that cover crops have are reducing erosion, reducing nitrate leaching, adding organic matter to the soil, and controlling weeds, pests, and soil moisture. Some farmers even let their livestock graze on the cover crops in the spring. In Iowa, winter rye is the most common cover crop because it has excellent establishment in the fall, good ground coverage, is very winter hardy, and can withstand those below zero temperatures that we have in Iowa. It also absorbs excess soil nitrogen and has a lot of biomass, as you can see in my pictures in the fall of 2019 and spring of 2020. The goal for the Iowa Nutrient Reduction Center is to have 12.5 million acres of cover crops planted in Iowa. But in 2018, there were only about 2 million acres of cover crops planted. So why are farmers not planting more cover crops when we see all of these benefits that cover crops have? Well, one reason is that winter rye, our #1 cover crop that we use in Iowa, is shown to reduce corn yield. Some possible reasons for this reduced corn yield is nitrogen immobilization, reduced water availability, allelopathy, and most importantly for us as pathologists, seedling disease. But how does seedling disease cause yields to decrease? Well, seedling disease causes damping off or death of the seedling, and this can reduce the population of the corn. It can also slow emergence, which reduces vigor. You can see there are several small seedlings, and they can be outcompeted for nutrients and sunlight by these larger seedlings. And then these smaller seedlings turn into weedy plants. And the weedy plants will produce smaller ears or be barren and have no ears. So this all of this leads to or contributes to reduced yield. How does rye cause seedling disease in corn, though? In our lab, we found that rye and corn are both hosts of some of the same pathogens. And the two most common pathogens that we found were Fusarium and Pythium. And of these, we see that Pythium was higher in corn planted into the rye, and Fusarium was not higher in corn planted into rye. So we believe that Pythium is the causal agent for seedling disease in corn when corn is planted after rye. We also found species of clade B and clade F were the most commonly found. Again, in this experiment, and clades are just groups of species that share similar characteristics. So clade B does well in cooler temperatures around 10 degrees Celsius, and clade F does better in a little bit warmer temperatures around 20 degrees Celsius. Pythium is an oomycete, sometimes called a water mold. It shares similar characteristics to fungi, but they are not true fungi. They're also soil-borne, which means that there are always populations of Pythium in the soil. Pythium has 3 different types of spores. The first is an oospore, which is a thick-walled, overwintering sexual spore. The sporangium is an asexual hyphal swelling that can germinate and infect the host, but it can also produce zoospores, which have flagella. And these flagella help the zoospore swim through the water to reach its host. So how does Pythium move from this rye into corn? In the fall, the cash crop is planted and the winter rye, the cash crop is harvested and the winter rye is planted and allowed to grow and establish. At this time, there is a living host in the soil for the Pythium to infect and multiply. In the winter, the shoots of the rye die off but the roots do not. So there is still a living host for the Pythium to inhabit. And the winter rye roots and the Pythium overwinter together. In the spring, the winter rye shoots continue to grow. And again, there's still a living host for the Pythium to inhabit and multiply. Then the winter rye is terminated, usually by spraying glyphosate. Once the rye dies, there's not a living host in the soil for the Pythium to inhabit, so populations of Pythium decrease over time, will eventually decrease unless we have another host for the Pythium to infect, which we do when we plant corn. And all of this is called the Greenbridge effect. So in the picture on the left, the arrow is pointing to the radicle, and the radical is the first to emerge out of the seed. Then we have these seminal roots around it, and all of these, this group of roots are the primary roots for the seedling until it reaches about growth stage V5 to V6. So damage to these roots can affect the growth of the seedling. And you can see in this picture, there's quite a bit of rot on the radicle, and there's even some on the seminal roots. And all of this root rot and damping off are symptoms of Pythium infection. So how do we manage Pythium in corn? Well, there are no corn varieties that are resistant to Pythium species, but we do have seed treatments. Metalaxyl and mefenoxam are the two more common active ingredients that are used to control Pythium. But seed treatments last only about 2 to 3 weeks once the seed is planted, and it takes about 2 to 3 weeks for the corn to emerge out of the soil. So once the corn emerges, the seed treatment is probably worn off, and the seedling is still vulnerable to Pythium infection. In our lab, we also did another experiment and found that planting corn 10 to 14 days after the rye is terminated reduced seedling disease in corn. So this is the typical recommendation that we give to farmers, but not everyone can wait this long. So we need to learn more about this system so that we can find other management practices. So we thought of this question: could social distancing rye from corn reduce seedling disease and minimize yield drag? So we hypothesized that physically separating corn from the terminated winter rye crowns will, 1, reduce the risk of seedling disease, and 2, benefit corn growth and development. So to test our hypothesis, I did 3 different experiments: a growth chamber trial, an experimental field plot trial, and an on-farm trial. So in my growth chamber trial, it was a pretty simple layout. I used field soil from a continuous corn field at the agronomy farm. So it was naturally inoculated with Pythium, and I didn't have to add more. We planted rye on one side of the pot and let it grow at 25 degrees Celsius for 30 days. Then we terminated the rye by spraying it with glyphosate. And 3 days after we terminated the rye, we planted 4 corn seedlings in the rye, or at 0 centimeters, or 8 to 10 centimeters away from the rye. We let the corn grow at 12 degrees Celsius for 16-hour days and 10 degrees Celsius for 8-hour nights. And these are similar temperatures to when we plant corn in the spring. They're also ideal temperatures for Pythium growth. We picked planting corn 3 days after the rye was terminated to be in that window of high Pythium inoculum. So when looking at seedling disease, we collected the seedlings around corn growth stage V1 to V2. We rated the radicals and seminal rot as a % area rotted. Then we looked at Pythium population densities in the radicals by extracting DNA and doing qPCR for both clade B and F. And we measured length of radicle in shoots, dry weight of the corn shoots, and emergence. So this is what we wanted to see. Corn planted into the rye would be more diseased and shorter roots than corn planted away from the rye with longer roots and less diseased, clean roots. But our statistical analysis did not show a difference among treatments for radical rot incidence and severity. But you can see that corn planted into the rye had numerically more rot than corn planted away from rye in both runs. Then if we look at seminal rot incidence and severity, we see there is a difference among treatments for at least one of the runs. And again, rot is higher in the corn planted into the rye than corn planted away from the rye. If we look at Pythium in the radicals, we can see that there was a difference among treatments for clade B, but not for clade F. And there was more clade B in the radicals of corn planted into the rye than away from the rye. And since we don't see very much clade F in my other experiments, I do not look at clade F. I only focus on clade B. Now, for seedling growth parameters, we can see that corn planted away from the rye emerged sooner, shoot dry weight was greater, radical and shoot length were longer than corn planted into the rye. So now we see that corn benefits from being planted away from the rye. So we moved this idea out to the field. And we did an experimental field plot trial out at the agronomy farm. And it was in a no-till corn and soybean rotation field. So our treatments are distances between the corn and the rye. And our first treatment is the control. Then we wanted to look at 0 inches between corn and rye, 7.5 inches between corn and rye, and 15 inches between corn and rye. And to get these spacings, we had different rye planting arrangements. So again, no cover crop, rye broadcasted, 7.5-inch drilled rye, and 30-inch drilled rye. So this is my field before the corn was planted. And broadcast rye is just basically rye sprinkled all over the ground. 7.5-inch drilled rye is 3 rows of rye with every 4th row skipped. And then this is what the 30-inch drilled rye looks like. And corn is typically planted in 30-inch rows. So the planter is basically just shifted over so that the corn is planted into these gaps. And that is how we created our distance between the corn and the rye. Some of the data that I'd like to show you for this experiment is rye biomass, seedling disease. I'd like to talk a little bit about what I found for my soil samples and just a few agronomic assessments that help answer the question, my first question at the beginning of this talk. First, I'd like to talk about weather, though, because it was very different for each year. In March, you can see that it was warmer in 2020 than in 2019. And there was more precipitation in 2020 than in 2019. And this helped with rye growth. You can also see that in 2020, it was a lot drier than in 2019. If we look, kind of narrow into around rye termination and around corn planting, we see that in 2019, it was a lot cooler than in 2020. And in 2019, we had more rain events than in 2020. And this higher precipitation in 2019 caused us to have to plant 8 days after the rye was terminated instead of the ideal 3 days after rye was terminated. But in 2020, we were able to plant corn 3 days after rye was terminated. Our rye biomass data are pretty straightforward. As spacing increased, rye biomass decreased. And in 2020, we saw a lot more biomass than in 2019. And this higher biomass is due to the warmer temperatures in March and more precipitation in March. If we look at seedling disease in some of my plots, we can see that in the control treatment, the roots look pretty clean and healthy. In the 0-inch treatment, the arrows are pointing to radicals with a lot of rot on them. And there's even one that is pretty short. In the 7.5-inch treatment, there is still a little bit of rot, but there are some seedlings that are clean and healthy. And then in the 15-inch treatment, again, they look pretty clean and healthy, similar to the control treatment. But when we look at the statistical analysis, in 2019, we did not see a difference among treatments for radical rot and seminal rot. But in 2020, we do see a difference among treatments for radical rot severity and seminal rot severity. And what we're seeing is that when corn is planted closer to rye, rot is higher. If we look at clade B in the radicals, in 2019, there was a little difference, and we saw that clade B was higher in the 7.5-inch treatment. Then for 2020, when corn was planted closer to rye, we see more clade B in those radicals. When I took soil samples to look at Pythium population dynamics in the soil, I had 3 questions. One, are Pythium clade B populations in the soil of the interrow, or where the rye was, greater than in the soil of the corn row? 2, are there differences in Pythium clade B among treatments? And 3, how do Pythium clade B populations vary in the soil over time? The answer to the first question is no. I did not see a difference in clade B from the corn row to the interrow. But we did see a difference for the other questions. I took soil samples at 3 different time points. When the rye was terminated, when the corn was planted, and when the corn was at growth stage V3, when we took those seedling samples for root rot. First, I'd like to point out that in both years, in the control plots, we see that clade B is pretty low in those control plots, and that is what we wanted to see. Then, so for our second question, are there differences in Pythium clade B among treatments? We did see, in 2019, we did see a difference at rye termination, and that is as spacing increases, clade B decreases. But we do not see the same trend for the other time points. In 2020, we see the same trend at all time points, and that trend is that as spacing increases, clade B decreases. So we do see a difference in clade B among treatments. And then if we kind of look at clade B in the radicals and in the soil, we see a similar trend in that so for 2019, we had higher clade B in the radicals at 7.5 inches. And we also have higher clade B in the soil at 7.5 inches. So that's pretty cool to see. And then 2020, we kind of see the same trend. So on to the third question, how do Pythium clade B populations vary in the soil over time? In 2019, clade B had a peak at around planting, and then it kind of decreased a few weeks later. But remember, we planted the corn 8 days after the rye was terminated. So that's also when we took these soil samples. In 2020, we do not see the same trend as we saw in 2019. Instead, we see that clade B kind of stays the same when rye was terminated to 3 days after rye was terminated. And then a couple weeks later, it drops off. And that could be because we only took these samples 3 days after the rye was terminated. And we might have seen a similar trend if we took these soil samples 8 days after rye was terminated instead of 3 days. So more sampling at more time points after the rye is terminated will help us understand how the Pythium clade B populations vary over time in the soil. Now on to seedling growth measurements. And these are from the seedlings that we took for seedling root rot. In 2020, it shows that corn planted closer to rye was behind in growth stage. In 2019, corn shoot height was greater in the 15-inch treatment, even better than in the control. So that's good to see. And then for both years, corn shoot dry weight was greater in the control and the 15-inch treatment. Chlorophyll readings and plant height were both taken when corn was tasseling. And you can see in this picture that there's a clear difference in greenness among treatments. And we can also see the same difference in our chlorophyll readings graph. So chlorophyll and plant height have about the same trend, and that is as spacing between corn and rye increase, chlorophyll readings and plant height improve. Now on to barren plants and yield. This is what farmers really want to see, and it's pretty straightforward. Again, as spacing between rye and corn increase, barren plants decrease, and yield of corn improves. So we took the similar treatments from the experimental field plot trial, and we asked a farmer, to help us out with an on-farm trial. And John Bakehouse in Hastings, Iowa, helped us out with that. So his fields were no-till corn and soybean rotation. Our treatments are very similar to that in the experimental field plot trial. And so we have a control, 7.5 inches between the rye and the corn, and 15 inches between the rye and the corn. And to get these spacings, he had different rye planting arrangements. So no cover crop, rye drilled in 15 inches, and rye drilled in 30 inches. So although we don't see a lot of rot or root rot in his fields for both years, we do see a difference among treatments, and it's the same. It's similar to what we've been seeing before, and that is that corn planted closer to the cover crop has more seedling disease or root rot than corn planted away from the rye. Same with clade B and the radicals. In 2020, we see there's more clade B in the radicals of the corn planted closer to the rye than planted further away from the rye. So for all of my other data that I took for the on-farm trial, I did not see a difference among treatments, even in yield. So this just lets us know that these results are not going to be the same in every field. And that's why it's really important to put these experiments in other fields. So our overall summary for all of the experiments is seedling disease is reduced when planted further away from rye. Pythium clade B populations in the soil were higher when corn was planted within rye versus corn planted away from rye. And increasing the distance between winter rye and corn reduces the negative effect of rye on corn and can improve corn yield. But these differences may vary depending on field and year. So back to the main question that I had, does the proximity of a winter rye cover crop from corn have an effect on corn growth, development, and seedling disease? And the short answer is yes. And now we have a new recommendation for farmers. So I'd like to thank everybody for listening to my talk. And thanks to everyone in my lab. They were tremendous help. And Keith Kohler, he took care of my fields and made them look all nice and pretty. And I think this was Tom Casper's idea. And all of my undergrads were a lot of help with sampling and data processing. And then Theo Gunther and John Bakehouse helped with my on-farm trial. So thank you everyone for listening. And does anybody have any questions?
Thank you, Sarah. That was a great job. We have one question in the chat from Aileen, which I can read. Are there any known effects of planting rye as a cover crop on soybeans?
In our lab, we did an experiment with that, and we did corn and soybeans, and we had rye before corn and soybeans, and then there was also camelina. And I think what we found is that rye doesn't really have an effect on soybean seedling disease. So yeah, I didn't really read too much into the results for disease on that. I just know that it doesn't cause as much disease in soybeans.
Sarah, can I ask a question?
Yeah.
So you said that you would be making a recommendation to farmers. What's your recommendation and why? Okay,
so I guess I would have several recommendations depending on what stage of farmer you're at with growing cover crops. So if you've never grown cover crops before, I would say try out broadcast in a small portion of your fields just to see what you find. If there's a difference between or a difference in yield between planting rye and not planting rye. But if you do have problems with that, I would recommend planting corn further away from rye or in these different rye planting arrangements. So for my experiment, it looks like planting rye on 30-inch rows is the best treatment if you are looking at disease and yield. So yeah, I would recommend planting rye on 30-inch rows to get that 15 inches between the rye and the corn.
Okay, thank you. Would you be coaching farmers that the results might not be consistent year to year?
Yeah, I mean, they're not going to be the same year to year. And I mean, that's just what we see is not every year's the same and not every field is the same. So,
But it's just so they don't expect a yield bump every
yeah.
year, right?
Mm-hmm.
Thank you.
I have a question, Sarah. So some of the earlier work that Allison and you guys have done in your lab was looking at the time of planting, right? So it may not be the social distancing spatially, but there was a distancing time-wise, right? So you did the planting of corn and that seemed to reduce the risk. So between those two options, right, the spacing of the rows of the rye and the corn, or the spacing in terms of time, I mean, how do you coordinate those two management options? It seems like there's two options there. Is there one that's easier for growers to do, or is there an advantage of or disadvantage of doing one versus the other option?
I don't really know about the ease of either of those treatments since I'm not a farmer, but the time one, I guess that depends on what the weather's like in the spring. And then for the space one, if there's not enough time, you kind of have to pick that in the fall. You have to pick whether you want to do that increased spacing in the fall. So I guess it's farmer preference, maybe. Yeah. Did I answer your question?
Yeah, I guess I hadn't thought of the timing. Yeah, the decision for the spacing of the rye has to be done in the fall.
So they could, this is just an educated guess, but they could pick the planting the rye in 7.5 inches with that gap, and then also plan to terminate the rye much earlier than planting the corn, just to have a safe, to kind of be safe, like you have the increased spacing if you need it, and then in the fall or in the spring, if the weather's not great and very rainy and you have to plant it close, that could still benefit.
Yeah.
I don't know if I said that right.
Yeah, rather than go with the 30-inch, go to the 15-inch as a sort of an intermediate option is already better. And then if they want to have extra reassurance, then delay the planting, but that year if they're able to do that, right?
Yeah, I'd still recommend delaying planting, just because then you know that Pythium populations are most likely going to be lower 10 to 14 days later. So yeah,
and maybe delay planting is not really the right term, right? It's more of the terminating early. You don't want to delay the planting of corn, but it's the rye earlier, so you have more of that gap. All right.
Yeah.
Hey, Sarah, good job. You mentioned the seed treatments available with seedling diseases may only last about 10 days, and that's about the time that corn is emerging. Are there any other chemical treatments available that could be applied like in furrow that could help last longer or help a little bit more?
I did read something about this. There are some in furrow treatments. I think I read it was xanthian. And that's also used for, to control Pythium. I don't know how effective or how common it is to use those in-furrow treatments. And I would be interested to know if the rye biomass or the stubble would interfere with that in-furrow treatment. I don't know, but yeah, there is in-furrow treatments that could be used.
What other questions do you have for Sarah?
Sarah, I've got a question. Another question, sorry. Why do you think the corn performance was worse at 7.5 inches? It was often worse than planting right in the row or right together with the rye. So what was happening there? I mean, 15-inch in the field gave you some moderately consistent, less Pythium, but what about 7.5 inches? Why did that bump up?
I'm not 100% sure on that, but 7.5 inches is still pretty close to the corn. And it could be that those roots from the rye, I guess they probably grew a little closer to the corn row, and there might have been more, and that would have given more inoculum, Pythium inoculum closer to that corn row. And I mean, there's still a lot of rye biomass in the 7.5 inch, pretty similar to the broadcast. So.
Okay, so you showed some data, I think, about biomass that was achieved by ryegrass in various stands. And if I looked at that graph right, it was about a 25% reduction in biomass broadcast compared to these spacings anyway. I mean, when you're talking to farmers about this, it seems like they're not getting the same benefit of the ryegrass if you're spacing it out like that. I mean, just part of the message, isn't it?
Yeah. You're right. The biomass is not as much if you space it out more. So that's one thing that we should look into, is how does that, I guess, less biomass affecting how the rye is benefiting the soil and like giving all those benefits. So we don't really know exactly if that less or that more spacing between is really helping this or benefiting the soil with reduced erosion, and nitrate leaching. I didn't look into that at all. And there's far less biomass in the 15-inch rye. So there's probably an impact from those treatments, or less of an impact, I guess.
Thank you.
Mark, and that's an excellent thing that you bring up, is that's one of the blind spots of the social distancing research, is that farmers are growing cover crops for the ecological benefits. And so, yes, if one of our recommendations would be to plant the rye in 30-inch rows, that means less biomass. And so how does that, as Sarah mentioned, affect soil erosion and nitrate leaching, etc.? So you'll be glad to know that that is a focus of a proposal we're working on right now. So,
and you didn't even have to pay me to say that.
Exactly. But no, you're absolutely, I'm glad you brought it up, because I was going to bring it up, that is a blind spot in the research. So
Thanks.
in terms of the systems research, right? in terms of the system. Any other questions? Well, thank you, Sarah. You did a great job. Really cool project, great data, and thank you all for attending. And we'll see you next week.
Thanks, guys.
Thanks, Sarah.
Good job, Sarah. Oh, she's gone.
Is she gone?
Yeah,
Okay.
she's like, I'm out of here.
No, I was waiting to see if she was going to stay on to debrief, but she didn't. So
Yeah, yeah.
yeah.
Probably
Yeah.
needs
I'm out of
to.
here.
I'm out of here, yeah. All right, well, we'll have more time to discuss things on Friday.
Okay. Thanks, Leonore.
Thank you.
Okay.
Bye.